Long time ago, when I was a graduate student, I got an idea that in order to improve water-soluble protein binding with the cell membrane, we need to modify the protein with fatty acid residues. And so I've developed a procedure for doing that then in inverse micelles, a field I was heavily involved in. And then someone learned about this technique from the paper and used it with the fatty acid shoots, the FAB fragments of the antibodies, and demonstrated that that results in increased delivery of the drugs to the brain. At that point, I got invited to an institute in the Ministry of Health, USSR, where I was essentially tasked to solve various delivery problems. And one thing which I was asked to do is we have these antibodies which get to the brain. Maybe you could attach them to the polymer and then conjugate the polymer with a drug. And so you can have a drug carrying polymer with the antibody to go to the brain. And it was seemed to be very complicated chemistry task, hard to control the polymer analogous modification. And I liked self-assembly. So we've done, instead of using these conjugates, we simply prepared the polymeric micelles out of Pluronic block copolymers. That's a trade name. They're known as poloxamers. So you take the micelles, put the drug in, and then you conjugate the block copolymer to the antibody. Now mix it all, and you have a decorated micelle, which gets to the brain. That was the very beginning of our work. About the same time, I was asked if we could deliver oligonucleotides. So what we've done, we hydrophobically modified oligonucleotides, and they were getting into the cell. We were thinking, how could we deliver the large DNA? Instead of chemically modifying DNA in order to make it more hydrophobic, we simply complex the DNA with the polycation, which contained a hydrophobic backbone or hydrophobic side chain, systems now known as polyplexes, and that worked. And then I moved to the cancer biology. And one thing we accidentally discovered was that our polymer, which we use, Pluronic, was hypersensitized in multi-drug resistant cells. And that's how we found the whole field of pharmacological effect of amphiphilic block polymers. And we worked a lot in this field, developing the first polymeric micelle drug to get into clinical trials, and also to develop formulations based on pluronics, which would inhibit drug efflux transporters in the blood-brain barrier and an intestinal barrier, and that was great fun. Then, speaking at Berlin at some conference, I met some scientists, Rainer Jordan and Robert Luxenhofer, two German scientists. They were working with polyoxazolines, and we had an idea whether polyoxazolines can do the same thing as Pluronic do. So we used them to produce the micelles. They were not sensitized in multi-drug resistant cells, but they were solubilized in enormous amount of drugs. That's how we developed the highly loaded polymeric micelles, and now embark upon the use of polyoxazolines and polyoxazines actually in various applications for drug delivery and for micelle drug delivery. And most recently, we started preparing delivery systems like lipid nanoparticles, where we replace polyethylene glycol with polyoxazolines and work in this domain. Speaking about the polyplexes, I realized early on that we cannot inject the cationic polyplex in the bloodstream because it's going to interact with serum proteins, stick to cells, and so on and so forth. And we introduced cationic block copolymers, which would bind with the DNA or oligonucleotide and form some kind of a polyion complex micelle. We call them block ionomer complexes. We've studied a lot the self-assembly of the block ionomer complexes for delivery of not only nucleic acid, but also proteins, and develop a variety of architectures which are currently actively explored and used by many scientists, such as cross-link polyion core micelles, such as nanogels, and the others. That is generally the recoup of what I've done in 5 minutes, and I hope this video is interesting to you.